Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 64th seminar, and I call it a University of Manchester Day. Last year, I took more than 160 days of trips to meet researchers all over the world, mostly in Europe. I'm especially happy to have almost 40% of this year's pioneer speakers outside the United States, mostly also from Europe. In fact, last week, we had one speaker from Belgium and the other originally from Israel. And today, the diversity continues, making this seminar series truly global. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Neil Dixon. He is a professor at Manchester Institute of Biotechnology in University of Manchester, UK, where I visited last year to give a seminar. I have known him for a decade through his publications, but I met him for the first time at a conference again in Portugal, Europe, last year, and then at his university later. He has contributed a lot to the scientific field of bioproduction and bioremediation, as well as my smile and laugh during our conversation with him, of course. And Neil, I really thank you for your time and contribution to the society, and the virtual podium is all yours. Thank you.
Okay. Wow. Thank you, Taysak. As warm and enthusiastic introduction as ever. I'm very flattered. Okay, let's see if I can do this pioneer title and status that you've afforded me some justice. Okay. Let's see where we're at. Okay then, so yeah, I'm going to share a few slides with you as part of this introduction to Yifan's talk, which is going to follow mine. And I want to try and be stimulating and inspiring and provocative. So yeah, if we look about our planet Earth, and we look at some of the challenges that faces our planet Earth, we can look at a whole range of issues, and you could argue that a whole bunch of these down on the bottom right are probably of political origin and have political solutions. However, many of the challenges that face us around the world have a biological origin and a potential for a biotechnological solution. As we know, recently over the last 2 or 3 years, the biotechnological implementation of the vaccine rollout has been able to scale at speed and at scale to address global pandemics. If we look, we cycle around anti-clockwise. If we look about ecosystem damage, we can think about ways in which we can engineer microbes and horizontal gene transfer elements, mobile genetic elements to address damaged ecosystems, to reverse damage that's being done to these, to remediate, to remove and degrade xenobiotics, for example. If we think about biotechnological solutions to climate change, there's those that are direct and indirect. Obviously, there's a big area within engineering biology, being able to make chemicals and fuels from more circular carbon routes from biomass and other based feedstocks, avoiding our reliance on petrochemical and moving towards circular bioeconomies. And more recently, much more direct work where researchers are looking to use engineered microbial systems to fix C1, CO2 directly to mitigate and hopefully reduce climate change potential. And then finally, obviously in the food crisis area, engineered microbes or microbes themselves, and then potentially engineered microbes have a big part to play in ensuring food security through nitrogen fixation and so forth, and helping to improve soil health and crop yields. So that's the area in which we sit, and I hope this is somewhere inspiring in terms of the challenge that we have and things that we can and should try to address. Obviously, these are technological solutions. A really important part of all this is to bring broader society with us to make sure that we're all educated and that we don't face the same and similar issues that we did around engineered crops previously, and make sure that we're all moving forward in the same direction. So there's a really big need to reach out and ensure that we're all developing these things together as a society. Okay, then I'm going to use the next couple of slides as a bit of a sales pitch to talk about who we are and where we're based. So my research group is based in the Manchester Institute of Biotechnology, which is based in Manchester in the UK. We're based in the Northwest, as you can see down here on this map here. So we're a part of the University of Manchester, but the MIB is a purpose-built research institute across many different faculties. It comes together, forgets about the classical disciplines and thinks about the ideas, the technologies and the solutions, really devoid of those classical disciplines. We're particularly known for industrial biotechnology, the production of chemicals through biocatalysis, although we have three broad research themes: producing chemicals and materials through biotechnology, overlapping with energy production, and more recently expanding into environmental biotechnology. There's also a big activity around medicine and health, both in terms of production of therapeutics, whether that's small molecules or biotherapeutics, but also into the sensing and diagnostics area. So at capacity, we're up to around 40 different research groups with just shy of 500 individual researchers that cross those different boundaries. Areas of expertise: biocatalysis, bioprocessing, directed evolution, synthetic genomics, structural biology, and data science, all underpinning all those areas are strong areas of activity. Whole bunch of major strategic investments have happened across the institute, both local investments and also UK government investments. It's home to one of the UK synthetic biology centres. This is the DNA or synthetic biology foundry. SynBYSS was formed to produce fine and specialty chemicals, and a whole series of analytics and robotics facilities underpin that. And that's, as I say, the national centre, and more recently the Future Biomanufacturing Hub, which is looking to scale technologies and push them up that TRL level and to overcome that valley of death as we push technologies outside the research base into SMEs and into industry. Okay, so that's the overview of the MIB. Just want to flag recently one slide in terms of self-promotion, in terms of who we are and what we do. So we're interested in trying to find biotechnological solutions to hopefully address some of the grand challenges that we face ourselves. We're interested in biorefinery and biotransformation, for example, how we can use renewable carbon-based feedstocks, particularly been working with groups out in Brazil to use lignocellulosic residues and take some of that residual waste material from that and make high-value chemicals through biotransformation and biorefinery. Using the same sort of concepts, we have also expanded more recently into kind of bioremediation, same sort of idea. Instead of funneling that carbon into central metabolism to make things, we're funneling to avoid it as a xenobiotic. We're particularly interested in plastic bioremediation, and we've done some structural biology and biochemical work, how the monomers that derive from PET plastic can be transported inside bacterial cells using one of these TTT transporters, using solid binding proteins here. So we did some structural biology around that. Really underpinning all of this is our kind of interest at the purely academic level around microbial gene expression. And we're particularly interested in developing genetically encoded biosensors. We've used transcription factors, ribo switches, and so forth to sense molecules that come from biomass, and also more recently derived from xenobiotics for bioremediation applications. In terms of optimizing these things, we use different statistical approaches, including design of experiments. And the final area, again, is to do with microbial gene expression control, but we're looking to fine-tune gene expression to match it to downstream capacity, ultimately to try to produce recombinant therapeutic proteins more effectively to drive down the cost of goods to allow more equitable global access to these kind of very expensive medicines. So in this particular example, fine-tuning in translation rates to match it to secretion capacity. But we're also interested in some recently observations where we can harness excretion, so that releases the protein into the extracellular matrix, which really facilitates that downstream purification process for therapeutics. Okay, so that's the pitch in terms of who we are. One final slide in terms of a call to action. One activity that we're also involved with is, no, sorry, two more slides. We're just flagging up some where this work has all come from and collaborators out in Brazil. So we've been working with Brazilian researchers for about 10 years now. We had a pump priming project back in 2014, and then a more recent project that's run for the last 6 years, looking around using metagenomics, enzyme discovery, lignin biosensors, and biorefinery to try to add value to the waste materials. I think Brazil is a really good example and probably the most advanced bioeconomy in the world, and they really have scaled their technologies with their first and more recently second generation bioethanol. So they're really good collaborators, and they really understand the scale and the scalability challenge as we look to commercialize biology. Okay, so the final slide then is just to flag up a dual PhD program that is between us in Manchester and Tsinghua University in Beijing. It's the first of its kind dual PhD program in synthetic and systems biology. I'm the director of it from the Manchester side, and George Chen, eminent professor from Tsinghua University, is the director in Beijing. We launched the program back in 2019 with our first cohort starting in 2020, middle of a pandemic, borders closed, so it's been somewhat challenging, but we have now just had our first crossover and exchange, so students have now been gone in both directions, but it's a joint program. Students are recruited from both sides, and they spend half the time in Manchester and half the time in Tsinghua University. So we hope this will develop further other synergies and collaboration between George's and his collaborators' labs and ours in Manchester. Okay, then, so that's a very quick overview of the challenges, the opportunities, Manchester, the MIB, us, and where we're at. So I'm going to wrap up at that point and then hopefully pass on to someone who's going to talk some more meat around modern science. Okay, thank you.
Okay, thank you. Amazing. You know, you and I share many vision, the same vision, I believe, you know, about almost everything, including the climate crisis and, you know, bioproduction. And that's, you know, another reason I really enjoy my visit, you know, your university at the time. And I now have new goal, because you mentioned multiple university. I only visited 3 of them, including you and UCL and Warwick. So this year goal is to visiting the rest of the university you mentioned. So also fantastic. I mean, I really impressed by your work in your university and some other people, and all the people kind of working together. And then I was also grateful because many people from your university actually joining this year's SynBYSS, you know, seminar series. So I'm really glad. Okay, thank you so much. I mean, this fantastic talk. I really enjoy. Okay, so now the main speaker of today with a little bit longer introduction. Dr. Yifan Dai got his bachelor and PhD degree in chemical and biomolecular engineering, both from Case Western Reserve University. At Case, he has been working on integrating synthetic gene circuit with electrochemistry for the development of universal biosensing systems. He is currently a postdoc associate at biomedical engineering at Duke. He currently works on engineering synthetic biomolecular condensates for cellular controls and utilizing synthetic system to investigate how chemical properties of biomolecular condensate enable condensate's cellular functions. He has worked on interesting interdisciplinary areas, and I am so excited to learn more from his talk today. And basically, his kind of interesting trajectory of career indicate how much he kind of learned new things quickly. And basically, he originally talking about during the PhD degree, electrochemistry, now combining that unique angle with the synthetic biology. So I believe he's one of the true leader in the future and then rising star. So I'm very excited again to listen to his talk today. So Yifan, thank you so much for your time today, and please take it away. It's all yours now.
Okay, thank you so much for the kind introduction. And you see my screen, right? Great. So I'm Yifan. I'm a postdoc at Duke Biomedical Engineering and co-advised by Toshi Cody and Ling Chongyu. So today I will tell you a story for how we can engineer programmable synthetic condensates for various kind of cellular controls. So if we come from the general synthetic biology perspective, we have two common goals. One is to have orthogonal central dogma, in which we want to design molecule de novo, and then to create a parallel cellular processes to the native cellular processes. On the other hand, we want to engineer capabilities to interface with current endogenous cellular processes to deliver new functions, the functions that benefit to biomedicine or general biotechnology. So the underlying core of two paradigm is that we want to know how to engineer in cellular functions. So what we have learned from nature. So nature utilized interaction to dictate function. One of the kind is lock and key interaction. So subspecific interaction leads to a binding formation of a complex, and the formation of a complex results in change of the cellular processes. But if we come more from the cellular perspective, cell is extremely messy, chock full of molecules, small or large. If you think from the protein perspective, every single protein move along its own diameter will encounter another protein. That suggests before a protein can dictate a specific lock and key interaction, they need to solve the problem in spatial regulation first. So instead, cell utilize the compartmentalization strategy to form certain kind of membrane-bound structures. For example, mitochondria. They have a distinct set of molecules that can dictate its energy production functions. But if you think a little bit more, what happened in the cytoplasm? It seems like an open space, but it have very distinct and also high fidelity cellular process undergoing. So question now becomes, how do cells coordinate biochemistry in a spatial temporal manner inside the cytoplasm? And one of such mechanism is to formation of a membraneless organelle. We also call it biomolecular condensates. So a subset of molecules that condense into a liquid droplet-like structures and to dictate function. The function include like granule formation, cellular signaling, stress response, even the organization of nucleolus. So I show you two examples today here. This is one of the first discovery for the underlying mechanism, which is phase separation, to show the condensate formation. Sorry. So it's look like an oil in water structure, but this is essentially water in water emotions. And this is our collaborator works on the formation of SARS-CoV-2 condensates, and we dig into the RNA grammar for how it can dictate SARS-CoV-2 condensates functions. So how it forms. Nature utilized intrinsically disordered protein to encode the capability to regulate function spatially. So it turns out more than 58% of a proteome contain intrinsic disorder protein, which is defined as no fixed tertiary structure, but have certain structure and symbol that can dictate functions. So if you take a more close look, if you think from even structure protein, before it starts to fold, it's a chain of polymers. The backbone is a chain of polymers. And the polymer can mediate this kind of weak multivalent dipole-dipole interaction. And by accompanying with cation-pi interaction, pi-pi interaction for short-range interaction, they can form this kind of multivalent and also percolated networks. So if you take a close look to the sequences, you will find like it's a low complexity, which means they have limited choices of the sequences to dictate this intrinsically disordered structures. For example, glycine or serine, they contribute to the solvation volume, and they also have what we call stickers, aromatic residues, or electrostatic interaction-based residues, arginate, glutamic acid, aspartic acid, to contribute this multivalent interaction as stickers. So phase separation process is always coupled with percolation. Either percolation happens before or percolation happens after. So it's a phenomenon-wise, it's a concentration-dependent process. Think from the cellular perspective. With increasing expression level, cells become more and more crowded. Once the concentration passes a threshold concentration, then phase separation occurs, forming a condensate and resulting in a second phase. If you come from the thermodynamic standpoint, so this is a phase diagram based on Foreign-Hugging theory. So temperature, pH, salt concentration, they can be triggers to phase separation. And then this is concentration. So above this binodal line is one phase regime that suggests it's no phase separation. Below this binodal line is two phase regime, so it's phase separate. So with increasing concentration, once you pass the phase boundary, the intermolecular interaction energy overcome the entropy of the system, results in like negative curvature of free energy, which corresponds to two distinct concentrations of the molecules. And then this leads to phase separation. So with increasing solution concentration, the dilute phase concentration and the dense phase concentration will now change. Only the volume fraction of the phases will change. So which suggests even you have a relative low solution concentration, you can have an extremely dense phase concentration. So this actually indicates how condensate can actually contribute to functions. So question for our engineer is more like, okay, how we can utilize this second phase in biology to contribute to control of synthetic biologies. Then if you think about this is a compartment, then we come from the compartment perspective, how compartment can dictate function. As I mentioned, this is a subset of molecule that can lead to phase separation, forming this kind of condensates. Then this can dictate selectivity by the type of molecules. Then if the reaction rate is solely depends on the concentration without the change of reaction kinetic constant, then increasing the concentration by phase separation forming the second phase can increasing the reaction rate. Second, then we have this inhibition function. Depends on what type of molecule we are recruiting into the dense phase, then we can protect the molecules. So this is how different compartment can dictate different kind of cellular function. So from our perspective, our lab interested in control of gene circuits. So we designed two different cellular functions as a target function to utilize condensates, and we try to think of the function that condensate can uniquely dictate. So one is plasmid sequestration. You think of a cellular population, every cell containing two plasmids, so two sets of genetic information. If we can form a condensate to specifically sequester one type of plasmid, then the following daughter cells upon cell division can possibly lose this plasmid. So we can control the gene flow, thereby regulating the phenotype of the cellular population. So this is more population level control. As I mentioned, condensate can form an extremely high concentration dense phase, and thereby we're thinking whether we can utilize this to further boost transcription capabilities. And question become how condensate can dictate such functions. So think about amber formation. Amber can preserve a material for thousands of years, and the key is to make an environment, a microenvironment that is distinct, that is very different from the outside environment. So we can do that in the molecular scale. So think of a condensate that need to arrest something. And you want the components to not free exchange with the surroundings. In the meantime, you want to design the pore size of the percolating network to be small. In the meantime, you want some function to have downstream impact, like transcription. And you want the condensate to be permeable. In the meantime, you want them to have high and rapid molecular exchange. And this leads to how we can design such modulus. So for DNA targeting, we use a DNA binding domain. Then to trigger phase separation, we use what we call synthetic intrinsic disorder protein, widely developed by the lab of Toshi. So they work on wrestling-like polypeptide or elastin-like polypeptide to make mutation to change phase boundary. But in my case, I select to change the patterning of the sequences. Thereby, I can manipulate the phase boundary. And further to increase the modularity and also control the cohesive interaction between the proteins, we add another dimerization domain as another valence for interaction. So we characterize the phase diagram, the material property of the condensates. At the end, we build mathematical models to connect how phase separation contribute to cellular process to understand different kind of C dilute concentration and the function connection with the cell with the phase separation system. So specifically for the design of synthetic IDPs, we first patterning the sticker. The stickers are the one that contribute to interactions. So this is a polyenphalate, and they have a homogeneous distribution of positive charge and negative charge residues. So the theory is, if we can segregation, like make it a block copolymer as one side of negatively charged and the other side of positive charge, we can increase the propensity for single chain compaction, which directly correlates with the phase separation propensity. So in our case, we start with a polyenphalate, which is a wrestling-like polypeptide, GRGDS-PYS. It have electrostatic interaction dictated by arginate and also aspartate assay. Then we utilize the tyrosine to dictate cation pi and pi interaction. Then by changing this polyenphalate, make a block copolymer with one side of the chain have positive charge, the other side of the chain have negative charge. We hope to regulate the phase separation propensity just by repatterning the key residues. Second, we learned from the theory that pi-based short-range interaction, either cation pi or pi interaction, they can dictate dense phase properties. So specifically, we're repatterning the aromatic residue into a cluster of patches. We can make super stickers, and such super sticker can change the condensate from this liquid-like spherical droplet into some like aggregation-like patchy structures. So in our case, we redesigned the distribution of aromatic residue by exchanging with the serines, which contribute to the solvation. Then we make this connected or different distribution as previous and make it more cluster, thereby enhancing the dense phase interactions. So based on these designs, we can actually have various kind of phase behaviors. So let's give the RD an example. So this is a cell concentration, as I mentioned, contribute to phase separation. This is like a driving force. And this is C light phase. It's a C dilute concentration. That suggests the phase boundary toward phase separation. So once we cross this line, this suggests it's going to become a phase separated system. So with the increasing salt, because of the electrostatic interaction, the salt start to screening out the interaction, thereby increasing the phase boundary concentration, which suggests decreasing the phase separation propensity. So basically lower the concentration suggests we have faster phase separation propensity based on the cellular expression level. So we can program this in various ways. In the meantime, it goes to the dense phase. The dense phase suggests how much protein is in the condensates. And we can further modulate different concentration inside the condensate. So this can be the foundation for how to regulate functions. We put it into E. coli first. So it's time dependent, which suggests it's concentration dependent after induction. And you can see with increasing time, it starts to forming condensates driving by wrestling like polypeptide, which is our synthetic IDPs. It can form distinct puncta. In the meantime, it maintain a dilute phase. The question now becomes, how does condensates can dictate cellular function? So we can form a synthetic condensate, but how are we going to use them? So what we learned from nature is that nucleocapsid protein, which is the SARS-CoV-2 condensate, that's responsible for viral packaging. So if you want to package something, you don't want them to lose outside. Thereby, the property of such condensate need to be dynamically arrested. That means the components inside the condensate can now free diffuse. So this is fluorescent recovery after photobleaching. So you have a molecule tagged with fluorescent labels, then you bleach to see how the dilute phase will diffuse into the dense phase. But you don't see any diffusion behavior for the signal recovery, suggests this is arrested. Then in the meantime, you have in nature, the transcriptional condensate is highly dynamic. Then you can see that for fluorescent recovery after photobleaching, once we bleach, it shows very fast recovery, suggesting the dynamic of the component can dictate the functions. So based on such, we build different kinds of synthetic condensates to characterize their physical properties. Specifically, with increasing dense phase interaction by one of the synthetic IDP, and we further fuse with the dimerization domain to increase the homotypic interactions, we can see that for fluorescent recovery after photobleaching, we show like no recovery at all. In the meantime, we characterize the pore size or the permeability of the condensate using a small dextron molecule. It's 40 kilodalton. It's fluorescent labeled. So you can see that this is a condensate, and the dextron now really partitions significantly into it with a partition coefficient lower than 1. But in the meantime, by decreasing the interaction, by removing the dimerization domain and also changing to a more soluble synthetic IDP, we can have full recoveries. In the meantime, the pore size we characterize by dextron for really big dextron, 150 kilodalton, it can actually partition in. So that suggests we have the capability to design and engineer different kind of synthetic condensate containing different kind of material properties. And also this kind of diffusion dynamics stay true in living system as we characterize in E. coli. So now question become, okay, how we can control functions. So the first application, plasmid sequestration to control gene flow. The logic is really simple. We have a cell containing 2 different plasmid encode 2 different antibiotic resistance. We form a condensate specifically target one plasmid, then upon cell division, the cell will lose. So it's asymmetric plasmid partition. So we design the circuits with a par B protein that can specifically targeting a par S DNA sequences. So with the formation of the condensates, it can result in over 92% of the loss of the cells carrying in 2 plasmid. And we characterize the phenotype by fusing a CFP onto the gene of the target DNA. And after a couple hours, we actually lost the most of the phenotype behavior of CFPs. So then we characterize this by different kind of synthetic IDPs. As I mentioned, synthetic IDP phase boundary can dictate when to phase separation. Then when to phase separation should equals to when to functions. So we actually find the kind of trend that we're looking for using different kind of synthetic IDPs. Increasing the phase separation propensity can actually increasing the functional efficiency. Then we model this by logistic growth model to trying to extract a parameter to see whether phase separation is really correlated with the biochemical parameter we characterize in vitro. So we use the artificial parameter alpha, which should be inversely correlate with the dilute phase concentration, which is the phase separation boundary of the system. So we have an estimate C dilute, and we have an experimental C dilute, but they are different components, but only based on the synthetic IDP differences. We actually find a linear correlation, suggesting phase separation correlate directly with the functioning efficiency of the system. Then this is a strategy to essentially control gene flow. So we were thinking, okay, what we can do with this. So in Lingchong's lab, he very interesting control horizontal gene transfer. So horizontal gene transfer is a fundamental mechanism by which the spread of antibiotic resistance happens. So how it works. So if the gene contains an origin of transfer, with the assist of F plasmid, it will lead to a conjugation process and move this gene into the recipient cell, thereby spreading the antibiotic resistance encoded in the gene. So our logic is really simple. We form a condensate to specific target the transferable plasmid, thereby inhibiting conjugation process. So indeed, with the encode of the sequences onto the plasmid, we can go over 1 order of magnitude decrease of the transfer efficiency based on horizontal gene transfer. So next, we demonstrate one type of the physical property contribute to condensate function. Then we were thinking, okay, whether we can control transcription by the other type. So if you think of different modes of transcription initiation, specifically in E. coli, sometimes we use T7 RNA polymerase, or we use the bacterial conventional RNA polymerase system. But both of this system utilize a single unit to initiate transcription leads to the production of mRNAs. Comparing with mammalian system, in which utilize multivalent interaction containing IDPs to mediate the formation of a transcriptional condensate, we were thinking whether we can utilize the same logic to boost transcription. So what we did is we fused a DNA binding domain with the synthetic IDP and utilize this binding domain to target the plasmid. So we want to recruit the plasmid into the condensate. In the meantime, we use the synthetic IDP to fuse with the SOX-S with mutation at R93A, which diminishes its own DNA binding capability. We want this to recruit RNA polymerase. Then we use this homotypic interaction between synthetic IDP to forming this condensate and recruit them both. So we utilize the gene circuits with the minimum promoter sequences, and then we hope this can boost transcription. So what we found is once we have every single parts available, all the components fused with synthetic IDP and also the functional domain, we can lead to the highest change of transcriptional signal once we induce the formation of the condensates. And more interestingly, if you take a look for the individual part, we can actually see the coalesce with this the DNA binding domain and also the SOX-S related fusion part, and also the RFP signal across the cells. And also we find the RFP signal actually has a high signal within the condensate, suggesting condensate really increasing the expression level. Furthermore, we were thinking, okay, what else we can do? So one thing we were aiming for is to program the hydrotypic driving force. The hydrotypic driving force means how many binding sites on the plasmid you can arrange. So what we did is increasing one DNA binding site from 1 to the 2. Then we can significantly further increase the RFP output as a signaling. So at the end, we were thinking, okay, our synthetic IDP really works pretty well. So we do some codon optimization. Then we put this into mammalian system to see whether we can control gene circuit, cellular circuit in mammalian cell. So we utilize the design from Xiaojing Gao's group at Stanford. So they have this citrine degradon reporters, which can be degraded directly through the temporal machinery in the cell. Thereby, this is a reported sequences will be degraded in the cytoplasm. Then we fuse this with the recruiting domain. And we also fuse the dynamic arrested synthetic IDP with also a recruiting domain to target this. So the logic is simple. If this is freely diffused in the cytoplasm, it will be degraded. But if this is recruiting into the condensate, we're trying to preserve the protein activities. So it leads to 25% of recovery, which is not really good, but it's really functional. Then we dig into why it's actually worked. So we find like most of the fluorescent signal actually directly only come from the condensates. So that really suggests condensates really preserve the material, protect them from the cytoplasma degradation cellular machinery. Then based on the same logic, we were thinking, okay, we can regulate protein circuits, any other things in the cytoplasma we can do. So it leads to translation repression. So this is not finished work yet, but I just want to show you how we can do this. We have a reporter gene based on CFP and fused with multimers of MS2 loop, which will interact directly with MCP. So we fuse the MCP with the same type of synthetic IDP that can lead to arrested condensates. Then with formation of the condensate, it can exclude the cellular machinery for translation. Then we can lead to repression of the expression level. So it actually works pretty well in our system for single cell characterization. It's very close to the negative control at the end. And if you take a look for what we took for fluorescent pictures, originally the synthetic IDP can only form this small condensates. But once we have RNA start to interact, it start to form this big condensates. So RNA can actually scaffolding the condensate architecture and make it look pretty cool. So with that, I want to summarize what I show you is that we show that how and to what extent such synthetic condensate can directly contribute as an orthogonal strategy for biological design. And one of the key is we utilize synthetic intrinsic disorder protein, which have really limited interference with endogenous IDP systems. Thereby, we can program various kind of functions. But in the meantime, we want to think about how condensate can be a unique capability other than current type of control to really dictate very unique functions through condensation. With that, I want to thank my advisor First is Linchong Yu here, and also Tosh Chikoti here. So I just get very cool advisor. They're extremely nice, and it's really guide me through all the journeys. I work with really great people within the lab for this work, including Donghan, Carrie, and Heiying, Xiao, Jonathan, Max, and Dan. So all my synthetic condensates were a collaboration with a bunch of pioneer people in the condensate field, supported by a Muri grant, including Rohe, Jose, Amy, and Cliff. So Professor Moon may recognize this is a worship. Okay, thank you very much for your time. And we all really appreciate Professor Moon for organizing this, and he really make a lot of effort to helping us. And thank you.
Oh, thank you. Amazing talk. Oh my God, I learned a lot today. And actually, I have some question, but before I'm asking question, I want to ask Neil, have any question or comment? If you want to, you can just comment or ask question.
Yeah, I've got a question. It's a really nice talk you found, really impressive. It's quite a technical one. I'm kind of interested in this kind of interplay between osmolyte concentration and condensate formation and dissolution, and kind of what's going on there. If I followed your data correctly, you were increasing the salt concentration, and that changed the speed of formation. I wonder if you knew what was going on there. Obviously, increased salt concentration actually shrinks cells. Is it that that's forming the increase in condensate formation rather than the salt directly?
So it's two different cases. First, it's what's the contribution of salt? So what we usually call polyenthylate. So it's basically you have positive charge, negative charge, amino acid residues. Then this kind of electrostatic interaction can directly contribute to phase separation. Thereby, we're increasing the system level of salt concentration. We are screening out the charges. Thereby, we're decreasing the phase separation propensity. That suggests the phase boundary is moving higher and higher because you need more protein concentration to trigger phase separation. So that's a way to understand the phase behavior of IDPs. Then in the case of the dense phase, it's a little bit more complicated. So how cell can contribute to dense phase is more based on the dense phase interaction. So I don't think we understand that really well now, but at least we know from the polymer perspective, it has salt in and salt out effect. So it's also have limited. So I give one example using wrestling-like polypeptide that's based on electrostatic interaction, favorable interaction overcome entropy, thereby phase separation. That's where the boundary come from. And then another case is called elaftin-like polypeptide. So that is fully inert hydrophobic residues. Then it's supposedly they don't form this kind of condensate if you think about they don't have interaction to happen. But that's actually driven by entropy. So with increasing temperature, for example, that's a LCST behavior. That suggests increasing temperature trigger phase separation. With increasing temperature, the water is start to move away from the backbone, increasing the translational entropy. Thereby, the protein itself collapse. So you can increasing in significant amount of salt. Then you can actually salt out the protein because the water is competing with the salt for the interaction with the protein. Then with the increasing concentration of the salt, you can further salt out the protein. So how is phase separation is purely based on the kind of the residues we have for the synthetic IDPs. And these two phenomena are also widely observed in native IDP systems. I hope this understanding answer your question.
Yeah, it does very extensively. Thank you.
So Neil, do you have any other?
No, that's very good. Is any more on the floor, as it were? I can always come back.
I have a question from Maya. I have to run to another building for class, but thank you so much. I love the synthetic ribonuclear protein granules regulate translation in living cell, and was going to ask about mammalian system 4 orthogonal processes, but it looks like you answered that. Can wait for the paper. Oh, okay. That's just comment.
Thank you.
Any other question? Okay, so in this I'll ask question. So I'm fascinated by the system, and I keep wondering during entire talk. I mean, to me, I mean, when I just heard the term condensate, I mean, I also heard that term before. To me, why biological system evolved to utilize those? I mean, to me, that's more likely, I don't know, I mean, some, response, I mean, some, respond to harsh condition like high salt or whatever.
Mhm.
But why even biological system, evolved to utilize? That kind of question kind of just lingering to my head, and I don't have any answer.
Okay, so I'll give you an example. So first is you think about cellular organization. It's not only the lock and key interaction is really important, which also contribute to condensate formation. But in the meantime, if you think from the cellular perspective, you actually need to organize the molecule into specific subcellular locations. And this kind of a needs for organization, which is what we call spatial temporal regulation, that is the way that they need to form certain structures or subcellular structure to dictate function. And from the functional perspective, it's kind of interesting to think about condensates. So condensates form once we pass a threshold. So it's a change of state. But for intermolecular interaction, dictate function, for example, RNA interference, you are expressing RNA inside the cell to inhibit gene expression, right? But if you use a condensate to inhibit gene expression, it actually can be possibly reversible because formation dictate function, which suggests reversible formation dictate reversible functions, which is not a simple capability can be dictated, for example, by RNAi, because you think about how you can control reversible cellular function. Thereby, condensates may provide a way to reversible control functional homeostasis within the cell for different purposes.
I see. That makes sense. But in that case, have you seen or read any paper that describe the enzyme condensate? So for example, what I'm thinking of, that's kind of related to my previous work, but that's a long time ago. But I remember, in mammalian system, there are some so-called scaffold that co-localize and then increase the final reaction rate, the flux.
Yes.
But I never seen anything related to those kind of thing in bacterial system that condensate basically contribute. But I just wondering, you ever seen any paper related to condensate that increase the final reaction rate because of the co-localization?
Yes. Our collaborator Jose Avalos, they have done this light-controlled metabolic system. So they utilize the synthetic system to trigger phase separation, like you mentioned, co-localization in certain. And they can increase enzymatic kinetics by increasing the local concentration. So the utilize of synthetic condensate to regulate enzymatic reaction is a pretty common trick people are doing in the field.
But my question is, do native system
I see. I don't
utilize the similar thing?
I don't really know.
Okay, yeah. But this is interesting. I mean, there are multiple different things we could do using the same concept. Yeah, I never think about condensate as a control system, but I should check out your paper after the seminar. That's a fascinating thing. Okay, I want to double check whether we have any additional question. Let's say, okay, the Maya's question is just already answered, so that's okay. And then I do not see anything in the chat. Okay, if that is the case, I will just conclude. Okay, so thank you all for joining and staying today. And we will meet again on January 26th Thursday, the same time, the same Zoom link, we'll have Professor Howard Salis from Penn State University and Professor Nicolas Sandoval from Tulane University. And as usual, the follow-up informal chat will occur without recording. And please stay here if you are interested in chatting with today's speakers, and I will promote you to panelists who can speak and show your handsome and pretty faces. And thank you, and I'll stop recording. Just give me 1 second. Stop recording.